This lecture talks about bone remodeling and the molecular mechanisms that are associated with it. First of all, we have to understand the basic details, such as the bone cells. There are three main types of bone cells: osteoblasts, osteocytes, and osteoclasts. Osteoblasts are young bone cells that are actively engaged in the synthesis of bone matrix. Okay, they add matrix, which is then mineralized, and in the same matrix, osteoblasts become mature and are now referred to as osteocytes. They simply remain in this matrix and maintain this matrix for some time. Osteoclasts are basically monocytes that have migrated to bone tissue and remain here, and their function is to dissolve matrix. We refer to that as resorption. Resorption is dissolving of bone matrix, and release the minerals, the calcium, phosphate, carbonate, etc. Okay, so those are the cell types. Now, there are many hormones associated with it. The action of parathyroid hormone in the regulation of bone resorption is important. There are many other hormones. Estrogen is one, calcitonin, growth hormone, etc. There are other factors such as growth factors, cytokines that play a part. Here we talk about the molecular mechanisms that are behind bone remodeling. That's OPG, RANK ligand, and RANK system. So let's understand this as best as we can. Osteoblasts express RANK ligand. What does RANK stand for? Receptor activator of nuclear factor kappa B. It's a mouthful, so you could simply abbreviate as RANK ligand. One thing I want you to pay attention to is the receptor activator part. So this is the one that activates a receptor called RANK, okay, which is expressed on the monocytes or the young osteoclasts, okay. If a RANK ligand binds to RANK receptor, then the cell that expresses RANK receptor will be allowed to differentiate or mature or survive and be able to do its function of bone resorption. Okay, so very simple. RANK ligand binds to RANK receptor and activates it to do bone remodeling. Okay, osteoclasts are the ones that express RANK, and therefore when RANK ligand is present and binds to RANK receptor, osteoclasts are able to survive, are activated, and they become mature cells. Okay, basically what happens is these monocytes are now able to differentiate, fuse to become young osteoclasts and then mature osteoclasts, and then they have expressed this, the structure changes. It's mononucleated to multinucleated now, and then their morphology changes. They have these folds in their cell membrane, and you could compare them to frills or basically the increased surface area to bind to the bone surface wherever they're present, and then release these acidic enzymes to dissolve bone matrix. Okay, so that's how RANK ligand, RANK receptor interaction leads to dissolving of bone matrix. All right, so I have shown here osteoclast expresses the RANK receptor, binding of RANK ligand to RANK triggers the survival of osteoclast, bone resorption. Loss of bone, too much of loss of bone results in osteoporosis. Basically, please note that the production of bone matrix and the resorption of bone matrix is a fine balance. Okay, the activation of osteoblast, activation of osteoclast must be maintained. And that is typically how bone structure is maintained. However, as a woman reaches the age of menopause, the decreased levels of estrogen, which is supposed to be protecting bone structure, is lost, and now the osteoclast activity is greater than the osteoblast activity, leading to thinner bone and leading to a pathological condition of the bone referred to as osteoporosis, porous bone, literally. So now another component that we need to talk about and its role is the presence of osteoprotegerin, OPG. So this is made by osteoblasts, okay? Again, a fine balance of the secretion of osteoprotegerin and RANK ligand is what is determinant in the healthy or not so healthy bone, okay? So let's see the action of OPG here. OPG blocks RANK ligand, okay? So the action of RANK ligand is controlled by OPG. So when RANK ligand doesn't bind to RANK, the cell survival is an issue. Apoptosis is cell death, okay? Cell death of osteoclast that would limit the action of osteoclast, decreased bone resorption. So less bone resorption in the presence of OPG. That's how you remember. Whenever you have OPG, RANK ligand function is lifted and cell survival of osteoclast is limited, and therefore there is less bone resorption. So the fine balance is now bone synthesis is greater than the bone resorption, okay? So no loss of bone, and that would be resulting in healthy bone, in other words. Let's talk about the role of estrogen. Estrogen stimulates the secretion of OPG. Estrogen stimulates the secretion of OPG. Estrogen also inhibits the expression of RANK ligand, okay? Estrogen stimulates the secretion of OPG, which should limit bone resorption. Estrogen also limits the expression of RANK ligand at osteoblast. I want you to also apply the function of parathyroid hormone. Parathyroid hormone acts on the osteoblast and stimulate the expression of RANK ligand. That is where it stimulates bone resorption. Okay, so remember, osteoblast is the target of both estrogen and parathyroid hormone. Parathyroid hormone acts in one way, that is by stimulating the expression of RANK ligand, whereas estrogen does the opposite, inhibits or limits the expression of RANK ligand. It also stimulates the expression or release of OPG. Okay, so in a menopausal woman, in the absence of estrogen, there is expression of more RANK ligand. All right, so all of this is important in understanding how bone remodeling is regulated. Okay, this is where we stop. Thank you very much.